Hello students, welcome to chapter 5. We're going to be talking about membranes and transport. So cell membranes, transport between the cell membranes, transport, yeah. Okay, first of all, the plasma membrane. So we know that the plasma membrane consists of the phospholipid bilayer, right? But it also consists of those proteins. So we have the proteins within that phospholipid bilayer, right? So these proteins are just staggered within it, just assembled within that film. So this plasma membrane separates the inside from the outside of that cell. Proportions of lipids and molecules depends on what the function of the cell is. So phospholipids have these sterols that will regulate how fluid that phospholipid is. And we'll talk about that here. So the phospholipid bilayer, we know that the phospholipid has a hydrophilic head, right? And the hydrophobic tails. This phospholipid bilayer will change depending on its temperature as well. So we know the polar end and the non-polar end and how it's constructed. But at low temperatures, the phospholipid bilayer freezes into a semi-solid gel-like state. So it'll kind of be more or less gelish, and you can kind of almost cut the layers in half. You can almost cut the layers in between those two phospholipid molecules. When a phospholipid bilayer is shaken in that water, you can form these small bubbles, these small vesicles, right? And so if you have a large bilayer and if you have a large cell or molecule, you can shake it and form these small bubbles, almost like soap. You can kind of shake around a large bubble and make it into smaller bubbles. Really quick, let's talk about these sterols. So cholesterol is important when we talk about the membrane as well. Membrane sterols have this dual solubility characteristic. Okay, so in a phospholipid bilayer, sterols align with the nonpolar interior, right? So it has that hydrophilic end and the hydrophobic tail, right? Just like that, just like the phospholipid molecule, right? So it has the same construction, so it'll align itself along that membrane, and the main sterols will help it maintain the fluidity throughout the bilayer. What do I mean by fluidity? So whenever you have a protein inside that bilayer, the proteins can go throughout the membrane. It's almost like if you we're swimming through water as opposed to like swimming through oil or swimming through jello. There's a fluidity throughout it, or a viscosity. So the more sterols you have, the more fluid these membranes are. So we have these membrane proteins, these proteins that I was talking about, that are inside the membrane. Transport proteins. These form channels that go in between the outside layer and the inside layer of that phospholipid bilayer. So they allow selected molecules, polar molecules, ions, whatever, to cross through that membrane. Right? So these polar molecules can be like water or nitrates or whatever. Okay, recognition proteins. These are proteins that can identify a cell as part of the same individual. So T cells in your body, these cells can attach itself to either a foreign object or a same object, and either identify itself as the same or as foreign. And if it attaches to a foreign object, it will, call other things over, and it will attach more T cells to that foreign object. And if a white blood cell comes through and sees the T cells, it'll attack whatever that foreign object is. So these are recognition proteins that are on the T cells. Receptor proteins. These recognize and bind molecules from other cells. So they act as chemical signals, such as hormones. So they'll bind to molecules, and sometimes those molecules will change what's going on inside the cell. So if a hormone is going past, sometimes it'll bind to that receptor protein, and it'll change what's going on inside the cell. Cell adhesion proteins. These bind cells together by recognizing receptors or other chemicals on other cells. So these will bind two cells together depending on what the other cell is doing on the other side. The fluid mosaic model. Overall, the plasma membrane is consisting of all of these things that I just talked about mixed together. So the phospholipids, the sterols, and the proteins, right? So all of that has to do with this huge mosaic within this phospholipid bilayer. Unsaturated fatty acid chains in the membrane. Phospholipids help keep membranes fluid at low temperatures. So at the regular temperature, you'll see this fluidity, just all those proteins keep it fluid, all the sterols keep the proteins moving wherever they need to go. At high temperatures, the rigid rings of the cholesterol help reduce membrane fluidity. So if you have high temperatures, the membrane reduces in fluidity, right? So there's this bell curve, right? At a certain temperature, it will stop fluidity. At a certain low temperature, it will stop fluidity. So mosaic refers to the membrane proteins, right? Most float in the bilipid layer. Some attach to the cytoskeleton. So the cytoskeleton is inside, right? Those microtubules, the kinetin, the actin molecules inside. Functional groups, active sites, face either inside or outside the cell, and then integral proteins are embedded inside the cell, while peripheral proteins are held by non-covalent bonds. So you'll have certain proteins that are just floating inside the membrane. Those are peripheral proteins. So we have evidence that supports this fluid mosaic model. Whenever I talked about the freezing technique, whenever researchers went through and froze the cells, what they did was they used a knife to cut open in between those two layers, right? And this is called the freeze fracture technique. So researchers split the bilayers into the inner and outer halves, exposing that hydrophobic membrane interior. They used electron microscope using individual particles the size of proteins. And they saw that they were embedded into the smooth layers. They saw that the proteins were suspended individually in the arrangement of like a mosaic. They were asymmetric. If they were symmetric, they would know that these wouldn't have to be clumped together, that they would have some type of focus. But they know now, or they know that using this technique, that they have a fluid motion. We also know that many eukaryotic organisms, they adapt to colder temperatures by changing those lipids inside of the plasma membrane. So this actually prevents the membranes from freezing at certain temperatures. Both the proportion of double bonds in membranes, so the double bonds in the fatty acid chain and the cholesterol content, that will increase at lower temperatures, giving the ability to stop them from freezing at that temperature. Now, if it gets too low, it's going to freeze eventually. So we have these transport proteins, right? Certain transport protein, or certain transport requires transport proteins, but not all of transport does. So we can talk about transport in general. So transport is the controlled movement of specific ions and molecules across a membrane, either by membrane proteins or just across a membrane in general. So we have passive transport, basically moving from a site of higher concentration to a site of lower concentration. This is moving across the concentration gradient through no energy, right? Doesn't require energy to do this. This is called basically diffusion, right? So there's simple diffusion and there's what's called facilitated diffusion. Simple diffusion requires no proteins, it requires no enzymes or anything. Facilitated diffusion does require a transport protein, right? So transport proteins usually have a larger central core that has the ability to transport larger molecules such as sugars, ions, amino acids, things like that. Simple diffusion is usually smaller molecules such as water, oxygen, nitrogen, CO2. And then we have active transport. Active transport goes from a lower concentration to a higher concentration. And this requires energy to do that, right? This requires energy to direct that flow, okay? And this also requires a protein to do it, an enzyme to do it, okay? Energy in the form of ATP. So this goes against the concentration gradient from a lower concentration to a higher concentration. So most biological membranes are selectively permeable. This means that they only select certain things to come through. So we know that hydrophobic or nonpolar molecules, you know what those are. These move freely through the lipid bilayer. Okay, this is passive, doesn't require energy. Hydrophilic molecules usually move through very slowly because they have to go through that hydrophobic core, right? So that hydrophobic core, the hydrophobic tails, usually will impede any hydrophilic molecules. So water, ions, things like that, they'll move rather slowly through there. Any charged molecules, charged atoms, things like that, are blocked by that selective permeability, are blocked by the phospholipid bilayer. Okay, this is important because charged atoms and charged molecules are very damaging to the inside of the cell. Okay, anything that is charged will usually react very heavily to any molecule floating inside the cell. Examples of transport proteins, so channel proteins, usual channel proteins, these are integral membrane proteins that form these hydrophilic channels, right? So if you have these hydrophilic barriers, you want the water to transport through because cells need water in order to function, right? And so you have channel proteins such as aquaporins that only transport water. So depending on the number of aquaporins in the cell depends on what the cell's function is, right? So if the cell needs a certain amount of aquaporins, or if the cell's function requires a certain amount of water, it'll have a certain amount of aquaporins around it. Another type of channel protein is an ion channel. This will be anywhere from sodium ions, calcium ions, potassium ions. These are gated channels that will only allow certain types of ions in and certain types of ions out. So one is a potassium sodium pump, So it'll allow one in and it'll allow one out. Gated channels, right, So it'll switch one open and then one closed, right? So the inside will remain open while the outside will remain closed, and then the inside will close and then the outside will open. Or it'll just open and close depending on what is going on inside the cell. And then we have passive water transport, right? Certain cells have just this passive water transport depending on what the cell's membrane is doing. So this is just the diffusion of water across this selective permeable membrane. So water just moves across it through a region of less solutes to the region of more solutes. Basically a region of higher water concentration to a region of lower water concentration. It's always going to be from higher to lower if you're doing passive water or passive transport. So movement of water by osmosis develops forces that can cause the cells to swell or shrink and shrivel. So either swell and burst or shrink and shrivel. All right, so you'll need to know these differences. So there's a demonstration down here, and it's this demonstration of osmosis, right? So glucose is in the solution. You have distilled water, H2O, right? It's just water. And if you put this glucose solution that's just full of glucose in the water, what's going to happen? Well, the water is going to go through that permeable membrane into the glucose solution. And the glucose solution is going to rise in the tube because the water is coming into that bulb. See? So the water is coming in and the solution is filling up. So that selectively permeable membrane is allowing water to come into that glucose solution. Basically raising the pressure, raising the water flow of that solution. Another way that you can see this is called osmotic pressure. And if the solution stops rising, then the pressure created by the weight of the raised solution, when it exactly balances the tendency of the water molecules to move from the beaker in the tube in response to the concentration gradient, that's when the osmotic pressure is balanced. That's what you call osmotic pressure. So when you have the right amount of water on either side, that's called osmotic pressure. So tonicity is a property of a solution with respect to this particular membrane. If the solution surrounding a cell has a lower concentration than its cell, the solution is hypotonic. The water will enter the cell and it'll swell up and possibly burst. So that means that the solution around it has more water in it. It'll enter the cell will swell up and it'll burst. Hypo, if you think about it, hypo, it's gonna well up, swell up, and burst. So if the solution surrounding the cell has non-penetrating solutes at a higher concentration than inside the cell, that means it's hypertonic, right? That means that water is leaving the cell and the cell will shrink. So it'll shrink and possibly collapse in on itself. If you have an isotonic solution, that means that the solutes and the solvent, usually water, is equalized between the cell and the solution around it. And that means water can flow freely between those two. This tonicity is very important when it comes to medical stuff. Like if you have a certain amount of salts in your system, you don't want to overhydrate, you don't want to underhydrate, because it can burst your blood cells. Or you can become dehydrated and you can become hypertonic, and it can catalyze your blood cells. This is especially true if you're taking care of plants. So in a hypotonic solution, we have these strong walls that prevent plant cells from bursting, right? So in animal cells, we don't have cell walls. But in plant cells, we have these really strong cell walls, right? So osmotic pressure in plant cells, this is called turgor pressure, right? This pushes the cell walls tightly against those cell walls, and it keeps their cells strong and supports the soft tissues, right? So you want a large amount of water within the vacuole. Isotonic, it actually becomes flaccid. It becomes almost withered. Wilted. So in a hypertonic solution, in extreme cases, the plant cells start to shrink so much that they start to become plasmolyzed. Plasmolysis, the cell plasm starts to break down inside the cells. So any amount of water in there will start to break down the cells itself. Okay, so there's different types of active transport as well. We have primary active transport. This is direct transport, right? So you're going to have ATP that hydrolyzes the power of transport directly. And then we have secondary active transport. This is an indirect hydrolysis. Basically, you have one protein that does a hydrolysis of ATP, and then another protein that has that same type of energy coming from the other protein. And that's the secondary active transport. So primary active transport, a sodium potassium pump. This is kind of what I was talking about before. So in the plasma membrane of all animal cells, we have some type of sodium that comes in, or some type of sodium that comes out of the cell, right? So 3 sodiums come out of the cell. You have some type of ATP, and then 2 potassiums come into the cell using the same pumping cycle. That's direct ATP driven. Gradients of positive ions established by primary active transport pumps underlie essential functions of life. So, this is a good amount of energy that we can get from just this system. Even though it does need energy, it does produce energy for us too. Calcium pump is another type of primary active transport. So this moves calcium from the cytoplasm, the cell exterior, and from the cytosol and the vesicles of the ER. Calcium gradient regulates the cell activities, such as secretion, microtubule assay, and muscle contraction. This is especially helpful in our brain. This calcium pump In muscle contraction, calcium pump releases calcium stored in the vesicles inside the muscle fiber, which initiates steps leading to contraction. So if you didn't have this, we wouldn't be able to contract our muscles, we wouldn't be able to pump our hearts, we wouldn't really be able to think. Secondary active transport. So pumps use an ion concentration gradient established by these primary pumps as an energy source. So if a primary pump is doing work, usually it has an ion that comes through and has some amount of energy to take it over to these other proteins to use as that type of energy. So that transfer is coming into these secondary active transport proteins. So the transfer of solute across these membranes is always coupled. Okay? So in symport, you're going to have a solute moving through the membrane channel in the same direction as the driving ion. Okay? So you're going to have two ions coming in at the same time. Right? So you're going to have one ion coming in from a high concentration to a low concentration. Right? You're going to have some type of passive transport, but you're also going to have the active transport coming in. You're going to have a low concentration to a high concentration. Right? So you're going to have a passive and an active going on at the same time. So symport going on at the same time, going in at the same way. Antiport means they're going in a different way. One's coming in and one's going out. So antiport, the solute and the driving ion are going in at opposite directions. So symport, same direction, antiport, opposite directions. All right, so on a different subject, we have pinocytosis. This is a cell's ability to intake an enormous amount of fluid. Okay, this is also called bulk endocytosis. There are a few cells that can do this. There's a few unicellular organisms that can do this. Bulk endocytosis takes in a drop of aqueous extracellular fluid that surrounds the cell. Bulk endocytosis sometimes, or pinocytosis, sometimes this is useful for feeding, but it's non-specific. It takes in any solutes present in the extracellular fluid because it lacks surface receptors for specific molecules. So sometimes the cell will just take in an enormous amount of fluid for non-specific reason. In phagocytosis, this is specifically for feeding reasons. Phagocytosis involves enzymes that breaks down materials, and then the cell sequesters those materials, those residues, to store in vesicles and expels them, or expels any waste product that doesn't need through exocytosis. Amoebas usually do this, so very small unicellular organisms, they can also do this. Euglena, they can do this. not Euglena, Paramecium can do this. For your attendance assignment, go through and name another unicellular organism that goes through phagocytosis. Just another type of organism that can do this process. Just look up on the internet and find one. Make sure to find a citation for it. All right, some type of citation so that I know where you would have found it. All right, and that is it. Thank you.